In this lesson, we're going to look at how living systems transport materials. The first aim is describe the processes of diffusion and active transport, then describe and practically apply the principle of osmosis. These are three types of transport. And then we're going to apply our learning of these three types of transport by explaining how water travels up a plant. Now, one practical application that science lends itself to very well is cooking. Believe it or not, studying science has made me a better cook. For example, take frying fish. If you've ever tried to fry fish before, you'll notice it's quite a messy job. Fish contain a lot of oil and they also contain a lot of water. And if you add small amounts of water to hot oil, you'll see that you get splattering everywhere. However, there is a simple trick to avoid this. Before putting the fish in the pan, if you salt them and leave them on the side for a bit, let's say for about 10 minutes, you'll see that water starts to emerge to the surface. Then if you take a paper towel and pat the fish to dry them off, you'll find when frying, without this excess water, you won't get any of that splattering. The fish will fry much better. The reason I'm talking about this is because this is a practical application of the principle of osmosis. Osmosis is how water moves from a high to low concentration across a semi-permeable membrane. Take this for example. Here I'm going to take an egg and I'm going to put salt on it. The yolk of an egg is actually a single cell, and like all cells, it's surrounded by a membrane. This membrane is semi-permeable. What that means, it has small gaps in it which allow small molecules like water through, but not bigger particles like salt. When you put salt on top of the egg, what happens is water gets drawn out of the egg yolk through the process of osmosis, and you can start to see the yolk get smaller. This is the same reason why putting salt on a slug kills it. So here you can see it gets smaller, and when you start to prod it, you can see it doesn't burst, it becomes more jelly-like and solid because the water is removed from the egg yolk. But osmosis is just one of three ways in which materials get transported within living systems. The first transport method we're going to look at in detail is diffusion. I've already talked about diffusion when looking at plants and looking at respiration. Diffusion is the random motion of particles from a high to low concentration. So particles will move from where they are bunched up close to each other to where they are not. Now this makes sense. So if you imagine these particles are moving around and where they're close to each other, you can imagine they're going to knock into each other. And when they knock into each other, they're going to rebound and start spreading outwards. What you will find is these particles will continue to spread out until they are evenly spaced within the area in which diffusion is happening. At this point, diffusion stops. So let's say someone's opened a bottle of perfume in the room. Now people sitting near that perfume bottle will smell the perfume first as those perfume molecules start to spread out, and eventually they'll spread out, evenly space themselves within the room so everyone can smell it. The other really important fact about diffusion is that it requires no additional energy apart from the kinetic energy already within the particles. So to help you understand this, imagine I had a tank of multicolored plastic balls. Now let's say I punch a hole in that tank. As you may imagine, those balls will now fall out effortlessly. They are moving from where they're in high concentration to low concentration, and I didn't need to add any energy to make this happen. This practical models diffusion really well. Now what we've got here is a chemical being placed here, and there's drops of a chemical on each little square here. The chemicals will evaporate just due to the fact that they're very volatile, and when they mix and react, there will be a color change in the drops over here. So we basically cover the squares so that the gases can't escape. And what you'll see is over time, you'll see color changes. So you can see these ones immediately around the chemical over here have started to turn black first as they react. Then if you give it a bit more time, you'll see that more and more start to turn black. So you can see these are the darkest because diffusion is happening closest to the source, but over time they will all turn black as the particles spread out and react with more distant spots. But why is diffusion important? Well, diffusion is essential for gas exchange to work in our lungs. So our lungs start off with a trachea which divides into two bronchi, which divides into bronchioles, and at the end of these bronchioles you'll find air sacs called alveoli. This is where gas exchange occurs. Every alveoli will be surrounded by blood capillaries, tiny blood vessels that transport materials. Now remember, we use oxygen in respiration, so oxygen is continually being removed from the lungs and taken to cells for respiration. As a result, there's very little oxygen present in the alveoli, but there's lots of oxygen outside in the air. This already sets up a concentration gradient for diffusion to occur. So let's imagine I'm breathing in. So now I've got my oxygen loaded inside my alveoli. In other words, there's a higher concentration of oxygen in the alveoli than there is in the blood capillary. As a result, oxygen diffuses into the capillary until there's an equal amount on either side. Now, if you also remember, during respiration, carbon dioxide is being produced. This is being transported to the lungs by the blood. So in the capillary, there'll be a higher concentration of carbon dioxide than there will be in the alveoli. Once again, this sets up a concentration gradient, so carbon dioxide will diffuse into the alveoli, where it will be in higher concentration than it is outside in the air. This again sets up another concentration gradient, so that when we breathe out, carbon dioxide moves out of the alveoli by diffusion. So now let's watch that a little bit faster. A similar thing happens when we look at gas exchange in leaves. Remember, in the palisade cells, photosynthesis is happening at a fast rate. This produces oxygen, so there's a high concentration of oxygen in the palisade cells than there are outside. So once again, a diffusion gradient is set up, and the oxygen will move out of the palisade cells through the stomata into the atmosphere. But oxygen will continually be made by the palisade cells during photosynthesis. Similarly, carbon dioxide is always being used up during photosynthesis, so there'll always be a low concentration of carbon dioxide inside the palisade cells than outside in the atmosphere. So once again, a diffusion gradient is set up, so carbon dioxide will move in through the stomata into the palisades by diffusion, where it will be used up during the process of photosynthesis. So remember, diffusion is the random motion of particles from where they are in high concentration to low concentration, and it requires no energy. Different factors will affect the rate of diffusion. For example, if you heat up the particles, they'll move faster and therefore diffuse faster. If these particles have more mass, they'll move slower, and that will slow down the rate of diffusion. Our second mode of transport is called active transport. The word active because it requires energy. You see, diffusion is a passive form of transport. It requires no additional energy. So active transport is when particles move from a low to high concentration, in other words, against the concentration gradient, using energy and a carrier protein. So this is what I mean by against the concentration gradient. This is not possible without energy, because you're basically forcing particles to where they naturally would not go. So if you take the plastic ball example, diffusion was just these balls falling out. However, active transport is the idea that we somehow need to get these balls back into this container. The only way I can do this is if I use my own energy to physically push these balls back into the container. This is what I mean by against the concentration gradient. So a really good example of active transport that you need to know is how minerals are transported into the root hair cells of plants. So root hair cells are specialized cells found in the roots of plants. They have an extension which creates a large surface area for the absorption of materials. Now here's the thing. In some situations, some materials are so useful to living things that we can't get enough. For example, glucose in our cells, we always need more precious glucose, and similarly, plants always need more mineral ions. So you can see we have this uncomfortable concentration gradient set up, where the green circles represent mineral ions. Now the plant needs to get these into the cell, but naturally this can't happen by diffusion because there's a higher concentration inside the cell than outside the cell. To help the plant do this, we need a carrier protein. That's what this is. This is a protein embedded in the cell membrane that allows things through. But to get the carrier protein working, we need energy as well. This energy comes in the form of a molecule called ATP, but you don't really need to know that for your exams. So what will happen is particles will move and bond temporarily with this protein. Energy will then be used to transform the protein shape, like so. This temporarily grants the mineral ion access into the cell, and then the process continues. So more ions will bond temporarily with this carrier protein, energy will be used to change the shape of the protein, and the mineral ion will move into the cell. So in review, active transport is the movement of particles against their concentration gradient from a low concentration to a high concentration. It requires energy and a carrier protein. So that's how we describe the processes of diffusion and active transport. Now let's look at probably the most complicated form of diffusion because it has the most parts. Osmosis. Now osmosis is the random motion of water molecules, only water. So if a question asks you how does water move, the answer will be osmosis. So the random motion of water molecules from a high to low concentration across a semi-permeable membrane. Now that sounds quite easy, but we have to understand what we mean by high concentration and low concentration of water to really understand osmosis. So imagine I have 3 beakers of water. In one of them I leave it as pure water, then I add a bit of salt or sugar to one of them, and then a lot of salt or sugar to the other one. Salts and sugars are dissolvable substances, so we call them solutes. Now, the pure water would represent an area of high water concentration, and the more solutes you add to the water, the lower the concentration gets. So this salt-filled beaker of water has a low concentration of water, if you like, because the water's in effect being diluted by the salt. So every centimetre cubed of water here will not just be 100% water, but also salt here, whereas here it'd be 100% water. So now let's understand the semi-permeable membrane. All cell membranes are semi-permeable. They contain gaps in them which allow molecules through according to their size. You can see that water can easily fit through, but solutes are too big and they can't get through the membrane. Then you've just got to work out where is the water concentration highest, on this side of the membrane or this side within the cell. Well, I've made this one pretty obvious. You can see lots of solute particles here and fewer here. Lots more water molecules here, fewer here. So obviously the water concentration is higher over here. So water will move in by osmosis, but it's a little bit more complicated than that. Well, it is if you truly want to understand it. Now, these water molecules are free to move into the cell, but also out of the cell too. So the question is, why will water end up moving in one direction? Well, it's to do with an idea called the sticky solute theory, and you will not need to know this for your exams, but I just think it helps you understand osmosis. You see, water molecules have a charge on them, and so do solute molecules, and they will attract each other. So when water molecules move and encounter a solute, they will stick to it, making the water stay on one side of the membrane. Of course, the same will happen on this side of the membrane too, but because there are more solute molecules on this side, the effect will be greater. Also, it's important to know that we're talking about the net movement of water when we're talking about osmosis, because remember, water is moving backwards and forwards. See, when I get paid at the end of the month, I get my net pay. That's my total earnings minus my tax. So basically, net means what's left over when you deduct something from it. So let's say in this example that 4 particles of water move through the membrane onto the other side, but let's say 2 particles move back. So we had 4 move that direction, but we had 2 move back. That means overall there's a net movement of 2 particles in this direction, because you just subtract that from that. But if you find that panicking, just remember most of what you need to know in a test is to do with this sentence here: the random motion of water molecules from a high to low concentration across a semi-permeable membrane. You just need to break down each part to understand it. So make sure that you know a high water concentration is like pure water, a low water concentration is one which has solutes in it, and remember semi-permeable membrane is a membrane or a barrier with gaps in it which allow small molecules through but not big ones. Or a simple way to think about it is water will move to where there's more salt and sugar, solutes collectively. You can investigate osmosis using the very tried and tested famous potato experiment. What you'll need to do is set up different beakers with different concentrations of water. Make sure they all have the same volume of water. So here I've got 3 different concentrations. You could be more accurate measuring them out exactly, but I'm going rough with this one. So I've got pure water here. Here I've got a very concentrated solute solution, and here we're going to assume that the concentration matches that of our potato. Because remember, inside a potato you have water, you have sugars, you have salts, and these particles give the potato its own concentration, and you could match that concentration by balancing the level of salt or sugar in water exactly. So the first thing you do is you cut multiple chips out of the potato, and you have to make sure they're the same dimension. I've got 3 chips in each batch for 3 repeats to make the test more reliable. I will then weigh the mass of the chips one at a time to find out their mass before submerging them in these solutions. After recording the mass of each chip before the experiment, I then submerge them into each of the solutions. And I could leave them in there for a few hours, let's say 2 hours. And what I do after 2 hours is take them off, shake the water off, make sure I pat any excess water off, and then re-weigh the chips and find out how much their mass has changed, and I could record that. But what's going on inside? You see, inside the chips we have lots of salts and sugars, so there's a low water concentration inside the chip. Outside, in the pure water environment, we have a very high concentration of water. Now remember, in osmosis, water will move from where it's in a high concentration to low concentration, so we'll get a movement of water into the chip. As a result, the chip's mass will start to increase as water moves inside the chips and they start to swell. So you could actually measure the mass, but you could alternatively measure the length of the chip as well, because as each cell fills with water, it elongates and stretches and extends the chip. The advantage of measuring length over mass is that you won't be misled by water content. You see, any water clinging to the chip will add to the mass, but it won't add to the length of the chip. Your choice. You could then find out an average of all the lengths or the masses of these three chips to get an average mass change in pure water. So now what's happening in the same concentration solution? Well, water may move in, water may also move out, but overall there'll be no net flow in one direction. There'll be no difference, so therefore there'll be no change in mass because we have the same concentration inside the chip and outside the chip. Whereas inside the concentrated salt solution, we have a higher water concentration inside the chip but lower outside because I've completely crammed this water saturated with salts and sugars. As a result, water will move out of the chip by osmosis. Notice how I say it. Water moves out or into the chip by osmosis, and the chip mass will get smaller. It could be a typical 2-mark question. Explain the change in mass of the chip in a concentrated salt solution. You would get 1 mark for saying the mass of the chip will decrease, because that's stating what has happened. But because it's an explain question, you need to give a scientific reason. And you'd say because water moves out of the chip by osmosis. You see, the word osmosis already tells you that water's moving from a high to low concentration across the semi-permeable membrane. So you don't need to explain that again because osmosis says that. However, obviously, if they're asking you to explain the process of osmosis, then you do have to say water moves from a high to low concentration across a semi-permeable membrane. So you could plot your results on a graph, and here you can see the graph has concentration in moles per decimeter cubed along the x-axis and change in mass along the y-axis. Positive above 0 indicating that the mass is increased, and negative below 0 indicating that the mass has dropped. So you can see at low solute concentrations, this time we're actually measuring the concentration of solutes in the water. So in other words, here is a low concentration of solute but a high concentration of water. We get a mass increase because water moves into the chip. So this region here represents when chips are put in concentrations of water greater than their own, or in other words, solutions of low solute concentration. At this point, known as the isotonic point, this is where the concentration matches that of the internal concentration of the potato chips. As a result, we get no change in mass. So this region represents solutions of high solute concentration or very low water concentration. As a result, water moves out of the chip and the mass decreases. So occasionally you get these graphs in exams, and sometimes it asks you, okay, so what about at this concentration? Predict what the mass change would be like. And all you have to do is extend the line downwards until it reaches that point, and you'd read off the graph to work out the mass decrease. So let's bring all this information together in a summary table. First up, diffusion. In diffusion, particles move from where they're in high concentration to low concentration. Diffusion requires no energy, it doesn't require a carrier protein, and doesn't require a semi-permeable membrane. Examples of diffusion are gas exchange in lungs and leaf. Active transport is when particles move against their concentration gradient, so from a low concentration to a high concentration. It does require energy, that's why it's called active transport. It does require a carrier protein, and it does not require a semi-permeable membrane. An example is mineral uptake in the roots of plants. This is a clever little stop-motion animation some of my students made to model active transport. Here, they're using a matchbox to represent the carrier protein. You can see that these are the mineral ions which are moving into the cell. You can see that there's more inside the cell than outside the cell, so there's a higher concentration. So these particles are moving against their concentration gradient. And you can see that incoming is an orange cube which represents energy. When it binds to the carrier protein, it changes shape to allow mineral ions into the other side of the cell. Finally, osmosis. Osmosis is the movement of water molecules only from a high to low concentration. It doesn't require energy, it doesn't require a carrier protein, but it does require a semi-permeable membrane. An application of osmosis is water uptake by root hair cells in plants. So once again, my students have created a different stop-motion animation here. These yellow squares represent solutes inside the root hair cell. These are soil particles, and you can see water's moving from a high concentration to a low concentration. This is a low concentration because of the presence of more solutes. And of course, you can see the semi-permeable membrane. So generally speaking, active transport is how mineral ions enter the root hair cell, and osmosis is how water enters the root hair cell. I have seen one fairly nasty trick that examiners pulled. They once gave you a picture of a root hair cell and showed that the concentration of mineral ions was higher outside the root hair cell than inside, and they asked how will the mineral ions get inside the root hair cell. Now, many students at the time had memorized that mineral ions, well, that's active transport. But they failed to notice that in this situation, we have a high to low concentration gradient. In other words, we don't need to push these particles against their concentration gradient. So the answer was diffusion. So generally speaking, yes, mineral ions do enter the root hair cell by active transport, but just be aware of the concentration gradient setup because it can switch to diffusion. However, if it's water movement, it's always osmosis, no exceptions. And that's how to describe and practically apply the principle of osmosis. So now let's tie in all our knowledge on plant anatomy and transport to explain how water moves from the root of a plant all the way up to the leaf. Remember, it's really important that leaves get water because only then can they remain upright and facing the sun. And they need to do that for photosynthesis. So if the leaf started to droop due to lack of water, it would no longer be able to photosynthesize effectively and generate energy for the plant's survival. Okay, so here's the first point at which you might slip up. You see, it's quite logical to assume, well, water's in the soil and it needs to get to the leaf. So you might think, I'm going to start explaining at this point here and work my way up. That's actually going to cause problems. Let's look at another example to help you understand. Imagine I was asking you to explain how the juice gets from the glass into the mouth through the straw. Now, you wouldn't start by explaining from this point. You wouldn't say, well, the juice leaps into the straw and then into the mouth. That doesn't make sense. Rather, your explanation would start at the mouth. Well, the mouth swallows air to create a suction. This creates an area of lower pressure here, so the juice is forced into the straw into our mouths. So it's very similar to that, really. The soil holds the water, which represents the juice in our glass. The xylem is like the straw, and the leaf is like the mouth, creating that suction to draw up the water. You see, there are water molecules in the leaf. Due to warm air passing over the leaf, the water molecules start to evaporate. In other words, they diffuse out of the leaf and get transported away by the air. This special type of evaporation that occurs from leaves is called transpiration. As water continually leaves the leaf through transpiration, it creates a suction, a vacuum, an area of low water concentration. So, water moves out of the xylem into the leaf to replace the water lost. This creates a suction at the top, an area of low water pressure, so the water moves in a continuous stream to replace it. This creates a vacuum at the bottom, so water will move in from the roots to replace the water lost in the xylem. As water is moving out of root hair cells into the xylem, that means there's a lower water concentration in the root hair cell. So, water molecules will move into the root hair cell by osmosis. So how do we write this? Well, firstly, remember we start with the leaf because the leaf creates the suction pressure that starts the process. Firstly, water transpires from the leaf. This creates a vacuum, drawing up water from the xylem. This in turn draws up water from the roots, creating a transpiration stream. That's a solid column of water that continually moves up the xylem. Then water moves into the roots by osmosis. Done. That could be a nice easy 6 marker. And that's how you explain how water travels up a plant.